Good morning, everyone. We will wait a moment or two as people join.
Morning, Lindsay.
While we're waiting for people to come on board, I want to make sure people are aware. An announcement has gone out through multiple channels of the annual FHS Brain Aging Program pilot grants. So I hope you have received that announcement at least through one channel. And consider applying. If you have questions, you should contact one of the two people listed in the announcement who are the directors of that program, Wendy Chu and Kathy Lunetta. Has anyone not seen that announcement? Because that will tell me whether we're getting the publicity out.
I didn't notice it, but
Huh.
you know, it's entirely.
I've seen it come out of the Andy Taylor's office. It's come out of the Evans Center.
Let's see.
ADRC.
In ADRC, it came out.
Okay, I just probably missed it.
I saw it.
Okay.
Okay, all right. So I think we have a quorum now. Welcome everybody to our monthly ARC seminar. I'm delighted to introduce our speaker for today, who is Rick Sherva from the Biomedical Genetics section. Rick has been a faculty member here for many years now, established a long track record in genetic studies of substance use disorders, but also has been toiling in the Alzheimer field for quite a while. And I believe he's going to be reporting on results from his studies looking at the contribution of viruses that are found in human DNA to Alzheimer's disease, and he has a NIH grant supporting that effort. So, Rick, the show is yours.
Well, good morning, everyone.
You're not in. You may need to switch screens. There you go.
That's not my first slide, however. Okay. I'll be presenting the work of our PhD student in the lab today, Marlene Tejeda, who's done most of this work. I've got updates on a couple fronts here. One, finally started to look at the genetics, and maybe the more interesting one is just things we've learned about the life cycle of especially herpes viruses themselves that partially came about through work we'd been doing, but some of the things I had just seen in the literature that I didn't know were true before. But I think a lot of you have seen, at least earlier iterations of this talk, but for those who haven't, I just want to give a little background on what we've been doing here. So, the idea that infectious agents could be Alzheimer's disease risk factors have been around for a long time. Viruses, bacteria, fungi, protozoans have all been linked to Alzheimer's disease in one way or another. You can see the references. A lot of these are through the same studies, but anyways, this idea has been around for a long time. The earliest evidence that this was Alzheimer's specific came from the fact that amyloid beta seems to have at least some immune functions. It forms globs around different viruses, including flu and including herpes. And if you stick a bunch of herpes virus in a mouse brain, it starts expressing amyloid. So herpes viruses are kind of unique in the viral world in that they can form these latent infections. Not a lot of viruses can do that. In their life cycle, I have learned a lot in the last few months about how these things actually work. You know, during acute infection, the virus actually gets into your cells, it gets into your neurons, it has its own, brings along its own viral DNA polymerase, RNA polymerase, it starts expressing viral proteins. There's these alpha, beta, and gamma that are early, middle, and late. Starts lyses the cell, more virus gets out, as most viruses do. Herpes has this unique ability that's kind of scary too. It can travel between cells without ever entering the intercellular space. Like it can form these capsids that can travel directly without being exposed to the immune system. But what makes it most unique is this latent period, which is mediated by this latency associated transcript. It's a little section of the herpes genome that is not fully understood. It acts in multiple ways to promote this latency period. So there's multiple different mechanisms through which this latency associated transcript works. It induces chromatin modification that alters, the availability of the viral genome for transcription. It directly silences gene expression through microRNAs, changes how the viral particles are transported in this trigeminal ganglion. I'm not sure if I'm pronouncing that right, but it affects apoptosis and lysis. And it's also involved in the reactivation. There are lots of different mechanisms for it to do that. And some people think it doesn't directly affect reactivation of virus at all, but it's, things like fever, stress, ultraviolet irradiation, abrasion, maybe Alzheimer's disease could be included there. But this is not really well understood. And this all came about for us trying to understand this very important aspect of this work, which is, the difference between latent and active infections. It's not maybe totally out of the realm of possibility that having a latent infection could be a Alzheimer's disease risk factor, but I think it's much more likely that, the cell death, the inflammation probably requires active infections, whether the number of reactivations is what's important, or whether the fact that they do it all. And these are all things we're kind of trying to figure out, and been thinking a lot about how through what we are doing, we could start to answer some of these questions. But this all started out when we saw this paper in 2018, where this Ben Reedhead did some really cool work where they took mostly RNA-seq data, again, mapped it to viral genomes, found a bunch of herpes 6A and 7 were associated with Alzheimer's disease, and they showed that the presence of these RNAs triggered expression changes in a lot of networks that have a lot of Alzheimer's relevant genes. So we thought, well, hey, we got a much bigger sample. We have DNA sequencing, but so we tried this in the ADSP data, and we recently had our contribution to this literature published in Alzheimer's and Dementia. So what we did that's a little different than what previous studies have done is, one, using only DNA, but we started by creating a new FASTQ file that only includes the sequencing reads that didn't map to the human genome. So once we made this new file, then we took 318 viral reference genomes that we got through NCBI and mapped them, mapped those reads to those genomes one after another. And we quantified those across within each virus and within each viral family. Normalized by the overall read depth. We think that would be important. We quickly realized that our results didn't make much sense. We were finding things like Ebola and viruses that are only known to infect corn smuts and crustaceans and things like that. So we went back through, or Marlene went back through and manually removed anything that wasn't known to infect humans and that wasn't acutely fatal, remapped it. And I think because of the fact that we're doing these sequentially and that there is so much homology across viral genomes, that once we remove those, almost all of them map to viruses that did make sense. And so we didn't know how well this was going to work, but I think we've shown that we are getting highly specific mapping, but that it is very much common in humans. So only 0.3% of the total reads we found didn't map to the human genome. So I think we would have been very concerned if, some or most, or I don't know how many we would have accepted, but if a lot of those were to map to a viral genome, we might think that would have just been a chance. But it turns out only 0.0001% of the non-humans read mapped to a different viral genome. But it was pretty common. 49% of brain samples and 59% of blood samples showed at least one viral sequence. I've shown these before. Here on the left is whole exome blood and then whole genome blood. But what I really want to show here is that the 80 cases and controls are separated here in pink and blue. The vast majority in both cases and controls and both blood and brain map to the herpes viridae family. And of those, the vast majority are human alpha herpes virus 1, the one that causes cold sores. And then of the non-herpes viridae 1, it's just a very small number. The next most common one is this papillomaviruses, which matches the epidemiological data. And we found a family of viruses that are fairly recently discovered, like in the 80s, called these TORC10 viruses. And nobody really knows what they do or if they infect humans, but they've shown that I should say they definitely affect humans quite commonly. They are not known to cause any symptoms or a phenotype or anything. Here that is in West blood and brain, just to show that it's the same across experiment type and across blood. So we recently, when we published this paper, we had about 17,000 whole genome sequencing samples. That's recently been expanded to 36 or 37,000. And I just wanted to show that the results didn't change all that much in terms of the proportion of each viral read that maps to these certain families. And same thing, herpes are by far the most common, and there are a few other ones that are detected with some frequency. So the sum total is we found 59 unique viral species. All of them were found in blood. Of those, 19 were found in brain as well. I don't see my number there. I think it was 10 that mapped that were specific to whole genome, and then 6 that were specific to whole exome. And then the total number is 10 that were found in blood, brain, whole genome, whole exome. And those are HSV1, Epstein-Barr, HSV6A, HPV6B, beta herpes virus 7, HPV71, Hep C, molluscum contagiosum, which is can cause rashes in kids usually. Again, those tortinomyvirus 9, and then tick-borne encephalitis. So to try and associate these with AD, Marlene ran a bunch of, I think, 7 different machine learning methods. She then took some of those and ran them through a logistic regression. We stratified by ancestry and tissue and by West WGS. And we also did the phylogenetic clustering. We're looking at families of viruses that are closely related. I've showed these before. Here are the machine learning results. The length of the bars here represent the number of different machine learning models in which that feature was selected as important. So we use this in addition to finding viruses that were associated, to
see which covariates we need to account for. You know, ApoE seemed to be an important predictor of AD, unsurprisingly, but we also found we needed to probably account for sequencing center and ancestry. I just wanted to show, I think it's on the next one. Maybe it was on the previous one, but we did find HIV in some of these. And I don't know if any of you have been HIV researchers in the past, but HIV should not, it's one of those pure RNA viruses where we should not have probably found any DNA of HIV. So anyways, I don't want to call into question my own methods here, but it was a little surprising that we saw that. It could have just been chance. But anyways, here's the results we published in our paper. Human alpha herpes virus 1 was highly significant. However, that effect was almost entirely driven by whole exome blood samples. We found some evidence for beta 6b, but it didn't survive multiple testing. This HPV 71 was significant. Again, this was driven by West blood, and then we found one African-American specific finding, that torctenovirus 10 that I was talking about, that people don't really know exactly what it does, other than that it gets into humans. So here are the updated whole genome sequencing results that reflect the 37,000 samples rather than the 17,000. That sample now includes a significant, small but not tiny proportion of people of South Asian descent from India, and also some Mexican ancestry samples. And so we were very happy to see what we did see with these. So rather than this all coming from whole exome blood samples, now when we do the meta-analysis of the whole genome, we find a highly significant association in African Americans. Also in European Americans, we find significant results with the whole herpes cluster, HPV 71 in European ancestry, also the cumulative viral load across all viruses. Of course, those are driven primarily by the herpes and the herpes 1.
Rick,
Yep.
can you comment on the odds ratios for HSV1?
I was just about to get to that.
That sticks out.
Yes, in the Caribbean Hispanics are mostly of Dominican descent. There's the most significant finding of all, and it looks protective. I will say that this is the smallest ancestry group of all, but I don't know what it is. I don't know if this is just chance or if it's something about the sequencing method or the sequencing center. But yeah, if this looks like if you happen to be Dominican, you should try and get Cold-Source, hepatitis C, and torctenovirus. I'm joking. I don't think that's really the case, but it's a weird kind of a thing, and we don't really know, other than the fact that it's the smallest ancestry group. If you just look at all the ancestry groups together, HPV 71 comes out, but again, protective, and cumulative viral load looks protective. But the fact that when your sample size increases, your results don't go away, and in most cases get better, was reassuring to us that our method was not just totally picking up on noise.
Rick, do you have Asians data?
We do. I'm not showing it here. We have the Korean samples that we're working on the viral detection right now in a bunch of Korean whole genome samples.
I see. Okay.
Rick, how many Dominican subjects did you have?
It's on the order of a couple thousand. Marlene, do you know right offhand how many exactly it is?
No, I don't remember. I'd have to check. I don't remember off the top of my head.
Yeah, I think it's a couple thousand.
That's
Right,
why
so-
that result with HSV1 is just so astounding. Not only because it's in the opposite direction, which explains why you don't see it in the total sample, but also the significance of it is such.
Yeah,
For a sample that size.
there are, I saw at ASHG a couple of years ago, there were people that were looking at I think it was based on medical records, but they were looking at herpes infections and cognitive performance, and they found a protective effect on cognitive function of herpes. Apparently helps you think better, is what they. So it's not unheard of that it could be protective in, if it's
But
activating your
maybe that gives us a hint about diversity and the differential kind of pathways that lead to disease among people of different ethnicities.
Yeah, I'm not entirely writing it off as chance. I think that's probably the most likely explanation, but who knows?
So Rick, this is a 70 kWgs, right?
No, this is the new 37K.
37K. So 37K should be roughly 6,000 to 7,000 Hispanic. Is this Caribbean Hispanic or Mexican?
Caribbean Hispanic, not the
Caribbean.
So one of the things I'm wondering is herpes viruses in general, I mean, if they infect discrete neurons, they're not widespread all over your brain. So it has to be the inflammatory response to the virus that's driving this, right?
That would be, I think, most people's hypothesis. Yes, definitely. I mean, they do kill neurons during an acute infection, which could be exerting an effect. They do get into the hippocampus.
So I just putting it out there for people. What if people looked at, I mean, are there transcriptomes available from either blood, brain, CSF from Dominican versus from Caribbean patients like at Miami?
Oh, meaning in terms of a reference genome?
Yeah, well, not just a reference genome, to be able to compare the transcriptomes of the people of Dominican descent or the Caribbean descent versus European, and then particularly look at the inflammatory pathways.
Yeah,
So data do exist. I know Richard Mayu has transcriptome data.
Yeah.
His Dominican sample.
So Rick, what if you went and looked at the cases that had the cases in Mayu's Dominican cohort that were positive or negative for HSV pathway, and then go and look at the transcriptomes, and in particular look at the inflammatory pathways. That would yield you, what's going on.
That is Caitlin Newman, who's on the call, another graduate student, PhD student in our lab, is going to do that.
Okay,
in
cool.
the samples where we have that, which would include the ROSMAP, and also I don't think that Mayhew's transcriptome data is publicly available. Wouldn't we have to get that from him directly?
Yeah, but
no, I know, it's just,
I mean, just reach out,
yeah, he's difficult to get data from, but
yeah.
We'll try.
Yeah, no, I don't have experience with stuff from him, so
yeah,
let's see, maybe you could put pressure on and help, or program officers could.
Yeah. So any more questions?
Sorry.
Yeah, nope. So one of the big mysteries that we still don't quite know the answer to is why we are finding HSV1 DNA in blood samples. It probably shouldn't be there. So first we thought, oh, maybe it's inserting itself into the human genome, and that, that it's part of a, one of the either white or red blood cells that we're getting in blood, but found out
that HSV1 actually does not acutely insert into the human genome. That doesn't mean it has at some point in the ancient history of primates. But what we do know is that HSV DNA is in the human reference genome. We figured that out because when we submitted our paper, one of the reviewers asked us to try a different software package called Kraken that can be used to map herpes or any viral DNA to the human genome. It was probably the person who wrote the software, but the difference between what Kraken does and what we are doing is it does not remove human reads from, your starting pool of reads. And when Marlene ran Kraken, we went from between 40 and 60% of the samples having at least one herpes 1 read to almost 100%. I think there were just a very few that didn't. So we thought, well, the only primary difference here is that it's not removing human reads. And the fact that so much went up, we just thought, oh, this has to be what's going on. I'm going to talk more about that on the next slide. But so thought, well, maybe we're just detecting active infections. It seemed a little improbable that 60% of the people in the sample had a active cold sore when the sample was taken. So we wanted to try and see if we could detect that latency associated transcript in the sample. So before, we weren't looking at where in the herpes genome our reads were mapping. So I was able to isolate that lat region and had Marlene look at that. And I think she only found 2 or 3 reads in the entire sample that we could map to the lat region. So, and the lat region, some people think it produces a protein, some people think it doesn't. That's not really relevant to what I'm saying here, but so there are papers showing that HSV1 is shed at very low levels during latency. We thought, the fact that we're doing PCR amplification, maybe all it takes is, one herpes virus in a sample that when that DNA gets amplified, that it's giving us enough to look at. Maybe it's something specific to AD patients where, that when they are producing low levels of virus, that it's getting out of the brain and into the blood. But the short answer is we really don't know. Our best bet is that low levels of virus are being shed even during latency, and that's what we're picking up on. But again, that's one thing we really want to look at going forward, is specifically looking at which parts of the viral genome are being mapped to, since we're finding the ones that are expressed during active infection. I guess if we're getting the whole virus, it maybe wouldn't matter, but I just want to know which of these genes, whether it's the early, middle, late, where we're seeing. So this is the herpes genome. Here you see the latency associated transcript RNA, got these early, middle, and late genes. So to confirm what we suspected based on comparing Kraken to our method, Marlene just blasted the herpes genome to the human genome, and sure enough, got a hit on chromosome 9. There are two regions that are 34 base pairs long. They each have 1 mismatch. So it's a pretty small section. It's not the entire herpes genome that has incorporated at some point in the past. I don't know if this is due to chance or what, but it makes sense as to why not excluding this human sequence would lead you to find herpes virus in everybody. I don't know whether it's relevant that it's a IL-3 regulatory protein that is proximal to this region. This is in a non-coding region, but there are genes obviously on either side of it. Okay, so the new results I want to show today that are pretty hot off the press. Marlene just presented these at ASHG last week, and so we haven't had a chance to fully explore these, but I wanted to show what we've done. So we wanted to figure out if there were genetic factors that are either driving people's risk to getting a chronic herpes infection. So to answer that question, we looked at viral quantity, looking at whether there's SNPs out there that are driving your risk of having a herpes DNA. I don't want to say infection because, you know, to be conservative, all we're really detecting, we can say, is that we're finding herpes DNA. So then, you know, included some covariates. We stratified by ancestry and whole genome. And then we wanted to know, you know, now that you do have viral DNA, whether there are SNPs that interact with that to affect your risk of AD. Again, we stratified this. Just going to say something else about this, maybe not. Okay, so we didn't find any significant main effects of herpes 1 on no genome-wide significant genetic risk factors for whether or not you have a herpes 1 infection. We did find significant hits for the herpes family, you know, all the sum of all reads in the across the herpes viridae family. This was specific to African Americans. Here are the genes we found. I'll talk about what they do in a little bit. We also found some SNPs that had an effect on cumulative viral load, not just including the herpes family. Again, these are all, it's driven by the herpes family, but here we found some additional genes. We first looked at all the known AD risk SNPs. And I was hoping, you know, since so many of those are involved in immunity and inflammation, that we'd find some specific hits that either interacted with or, you know, had a main effect on your risk of having the virus. Of those 83 or 86, only one showed a significant effect. This was in whole genome sequencing samples in a trans-ethnic meta-analysis. This is TPC1, and of all the, you know, third of all AD risk genes that are involved in immunity inflammation, the one we found was not among them. So I don't know if I'd say disappointing, but not what we expected. Oh, sorry, 83. Okay, so for the interaction tests, we only found 3 significant interactions between, this is cumulative viral load and SNPs. This is, again, a trans-ethnic meta-analysis of all whole genome sequencing samples. There's this Claudin 19, which is a transmembrane protein that controls tight junctions. It looks like it has immune function. This middle one is not near a gene that looks anything like it would be interesting, but this other one out on chromosome 12 is near this SUDS3 gene that maps to the Go term infectious disease. Here are those 3 SNPs. I just wanted to show that that we're seeing a significant effect direction in most cases across the various ancestry groups. Here, the only one that's in the opposite direction is in Framingham, which I guess I'll mention that that's the only one where we did not split that one out by ancestry. The non-European sample size in Framingham were so small that our results were just all over the place when we tried to stratify those out. So we just ended up running those all ancestry groups together and adjusted for principal components. Seeing for this first one, you see a pretty strong effect in AAs, you know, anomaly significant in EAs. This is the Mexican population, not significant in Indians, but has a significant effect in Caribbean Hispanics. And the Caribbean Hispanic is in a consistent direction, which, you know, again, I don't know what to make of that, given the main effect on HSV1 on Alzheimer's disease risk is opposite there. Same here, driven by African Americans, Europeans, and Mexican. This, again, has a signal in Caribbean Hispanic. And then that first one
you can see. So nice to see that for most of these, there's contributions across different ancestry groups.
Rick, can you comment on the minor allele frequency differences?
Yeah, I couldn't fit it on here. The smallest one of these in any group is 7%. They're pretty common, and they're not the same allele frequency in all the different ancestry groups, but they range from 7% to 23%. So just mention what some of these genes do. So of the main effect ones, there's several of these that seem to have either roles in immunity or cognition, you know, this KTMA2 memory formation, synaptic transmission, excitatory neurotransmitter. This one has actually been proposed as a plasma biomarker to discriminate AD cases from controls. They're involved in innate immunity, so that's definitely probably our number one candidate. CLEC12A regulates the expression of interferon I. Inflammation response to cell death, that's a very cool one too. This one is directly involved in antiviral response, susceptibility to flu, pleiotropic cytokine, adaptive immune tolerance. So found some very interesting genes in African Americans in terms of having a main effect on either herpes family or total viral load. So I'm almost done here. Our first thing we want to do is accumulate more data. Younga and Xiaoling and I recently bought the Cadillac access to the UK Biobank data, which means we can access their sequence data. We don't have that access yet, but when we do, as most of you may know, if you're going to do things like this on the UK Biobank sequence data, you have to do it on their computers. And I just don't know whether somebody would be willing to, run this for me, or if it would just cost $100,000 to buy the compute time to do that. But to be able to do this on that many samples would be very cool. We're also, the wheels have started turning to potentially do this in the Million Veterans Program, whole genome sequencing samples, which are over 100,000. So potentially we might increase our sample size by several orders of magnitude in the coming years. We still want to get at this question of, whether these are cause or an effect of AD. It's certainly not beyond the realm of possibility that, when you get AD, you're more likely to have active infections, or, your immune system starts to go. So this is one of Marlene's thesis aims, is to try to do Mendelian randomization to try and establish some causality, and the running the genetic association studies was the first step towards doing that. Both Marlene and Caitlin are going to look at not just AD itself, but if we can link this to different aspects of AD pathology in terms of imaging or cognitive decline or biomarkers or expression, that would be very good. And I don't know that doing case-only analysis is going to be feasible for these endophenotypes. Obviously, we have a lot of cases, but if we could show differences among AD cases and AD cases in terms of AD pathology based on whether or not they have the virus, I think that would go a long way towards showing that these are, in fact, causal, or at least changing certain aspects of AD pathology. But the number of case samples that have these endophenotypes and imaging things is pretty small, but we're going to give it a try. I really want to see which parts of the HSV1 genome we're detecting, if there's differences in the, if it was spread evenly across the herpes genome, that would, I think, indicate that we're just finding the whole virus. Whereas if it were certain parts of the herpes genome that are active during active infections, I don't know what that would mean, but it might mean that we are detecting active infections rather than just random virus that's floating around not doing anything. Then again, we got to follow up on the GWAS results. Those are pretty new, and we've got a lot of post-GWAS bioinformatics things that we're going to do to try and dig more deeply into those. And that's it for me. I just want to acknowledge the people that have contributed most. I guess I'm going to have to add Caitlin to this slide. Sorry, Caitlin, it's old. So yeah, that's all I had. If there's any questions, I will take them.
All right, we'll open up the floor for questions. Anyone want to shoot one at Rick?
Rick, I remember last time when you presented, you show ApoE4 carriers and non-carriers have a different viral load of herpes virus. These data still hold when you use large?
No, that's one of the things we have to dig into more, but no, neither in the main effect model or the interaction model did the ApoE SNPs themselves turn up. We have not looked at the actual E4, E3, E2 serotypes, or so it is just the SNPs, which in any AD analysis come up as very significant. So I don't think we're going to find an ApoE effect, which I kind of thought we were.
Okay, and in different ethnic group, it's still the true, right? You don't see ApoE genotype effect.
No.
I see. Okay, thank you.
Carmela has corrected me that HIV is, I thought HIV was a pure RNA virus that does not produce RNA.
Other questions?
Rick, this is great. I may miss something. Did you look at the sex effect? Sex is stratified, or the sex has any different impact in viral load or anything like that?
We did not
herpes virus, yeah.
So I mean, we did not look at whether sex affects virus. We know sex had an AD effect, so we adjusted for it in everything we've been doing. But no, I did not. I don't think, Marlene may correct me. Did we look at whether sex predicts virus?
I haven't done it recently in the new GWAS studies, but previously when I ran with the whole exome 20K, I noticed that there was some sex differences, specifically in females. But it's been a while since I've looked into that.
We could just pull up, you could just look at the sex term in your main effect GWAS models to.
Yeah, so what about sex stratified? I'm kind of curious. Sex has some people huge impact on the virus, microbiome.
Yeah, especially with the HPV finding we have. Yeah, we'll have to dig into that.
So Rick, I've got a couple of questions that are related to the population differences in your findings. So, of course, the most startling one is the one I mentioned while you were presenting, is the effect of HSV1 in the Caribbean Hispanic group is not only in the opposite direction, but in a comparatively smaller sample, so significant. So my question is, do you have thoughts on how you're going to try to follow that up?
Yeah, in these new analyses, I don't think we've looked at the viral distributions by ancestry group. So I think that would be the first place to start, to actually look at the proportion of Caribbean Hispanics who have the virus and the quantity, and compare that to the other population groups to see if it matches the other ancestry groups and still has a protective effect. That would make me think it was real. But if
you might consider, if these are Caribbean Hispanics, right?
Yeah.
So there may be a point where you might want to reach out to Richard Mayu and get his insight input here.
Yeah, I will do that. I'm hoping this might interest him enough to maybe speed up his generally glacial pace of sharing data with anybody.
If not him, there are other people in his group who might be very interested, Giuseppe, for example. And the other related question is, I'm trying to refresh in my mind the findings where you had, I guess it was from the interaction GWAS, and you had 3 top genes, and you showed the results within each of the ethnic groups. And if I recall, not sure if it was for all of them, but most of the evidence was supported by the African American, Caribbean Hispanic, and to some extent the Mexican.
Very nice.
Any thoughts on what might be going on there, especially since the European ancestry, I presume, is by far the largest sample?
Yeah.
Very interesting.
You know, another thing we haven't dug into is what, for these interacting SNPs, what the main effect is. We don't know. I haven't looked yet at what that interaction looks like in terms of, you know, whether it's opposite directions or just different slopes. So I think looking at the main effect of those SNPs within each ancestry groups is probably the first step.
Yeah, and again, certainly what they have in common, the Caribbean Hispanic have a high African ancestry. I don't know about the Mexican Americans. I think they have a higher Native American component. I don't know their African component. Other questions or comments from folks?
Rick, I have a question. Herpes infection is really, I think, different among different age. If you do older population, probably those herpes virus has been sitting in human genome for a long, long time. And if you, because you have large cohort, did you ever do stratification of age? And then you maybe GWAS will show different.
Yeah, so the age range of this, you know, these are all elderly cohorts, Alzheimer's cohorts. So you know, minimum age is probably 60 or 65.
Okay.
But, you know, we do run stratified GWAS for Alzheimer's itself, which isn't to say that there couldn't be effects in, you know, the 60 to 90 range.
I see.
You might consider running an age interaction term.
Yeah, that could be informative, definitely.
Yeah, hi, Rick. I guess my question is also related to Wendy's question. So if I got right, the virus load are inferred from the exome sequencing or whole genome-wide sequencing data. And that kind of thing could be changing like during the lifespan. And I guess for FHS data, and maybe the sequencing, is it coming from little earlier sample collection? And would that be, you found would be different from like older Alzheimer's disease related cohort?
Yeah, so the age that we have is, I believe, when the sample was taken.
Yeah. Oh, I got it.
Is that, well, Rick, the ages in ADSP data sets tend to get updated based on their last clinical exam. So they're not, some may be coincident with the age of blood draw, especially if they were seen only once. But they're, the age at which the blood was drawn for many of them could be several years earlier. I'm sure for almost all of them, it's at a late stage in life. So you're talking 60 versus maybe 70, someone's designated age based on last exam, and the blood might have been taken 10 years earlier.
Yeah, I guess that's especially relevant in Framingham.
Rick, because you just said you're going to get UK Biobank data, and they have large age range.
Yeah, we would probably be limited, for AD based analyses, we would be ignoring the younger people. I would probably still look at their virus, profile just to have it. But yeah, in terms of AD specific analyses.
Yeah, Rick, just curious about technical issue. In yesterday's ATSP biomarker meeting, they mentioned, especially compared to tau, amyloid beta is easier to be not contaminated. If the blood plasma collected much early, store much longer time, and the variability for the amyloid beta is large. So similar here, if the blood draw, blood stored very long time before you do the DNA sequence, is that will be affect your result?
It very well could. I don't know if I don't know that viral DNA would, react to time any differently than human DNA would. But was what they were talking about specific to DNA or just the biomarkers?
They talk about biomarker yesterday, yeah, which means the plasma, when you store plasma in -80 freezer long time, compared to recently collected, that will affect your amyloid beta more than for tau measurements biomarker.
But those are measuring levels of protein.
Yeah. Just I'm
DNA.
curious. Yeah, I'm curious whether for the DNA. For DNA, we know if you DNA collect from brain, brain is has more contamination compared to DNA from blood. But I don't know the time when you store in your -80 refrigerator. Yeah, before doing that. Just
I've never
curious.
heard what.
Well,
I've never heard whether.
Actually, if Carmela is still with us, she can recall the experiments we did. I don't know, about 8 years ago, when we were growing up cells from frozen, that were frozen from back in the 90s, early 2000s. And then 15 years later, we were growing cells and looking at levels of A beta and tau with respect to the gene we were after, ACAP9. And it didn't seem to impact the ability to measure and see results.
I guess I haven't heard whether.
Comments for Rick? Or Rick, do you have any other comments?
No, I was just, I never occurred to me. I don't know whether the length of time a sample was stored affects whole genome or whole exome sequencing significantly.
I wouldn't think so, unless it's the integrity of the DNA, but shouldn't change the sequence. Well, if there are no more, go ahead, Lei.
Yeah, Harry, I have another question. I mean, so how likely the virus genome can also get epigenomically modified? So which means using the data from blood trypsin data, how likely we could maybe identify the virus genome? Is
I,
that
I
possible?
missed the point of the. Can you say that again?
So how likely the virus genome also get epigenomically regulated? And is that possible?
Oh,
Lipstick data from blood, we also could capture something. Like
yeah.
usually people do that and then you map to the human genome, right? And I was wondering by your talk, how likely maybe there is something also could map to virus genome.
Yeah, I don't think epigenetic modification would affect the mapping quality of the reads to the viral genomes.
Okay. Maybe it's worth trying. Thank you.
Okay, well, if there are no more questions, I first want to extend a big thank you and congratulations to Rick, Marlene, and Caitlin, who all worked on this project and are showing results that clearly are still in progress, but very exciting. Great presentation. And I know since our group here has increased since I gave opening remarks at the beginning of the hour, I just want to remind everyone, hopefully you received the announcement from some channel about the FHS Brain Aging Program pilot grants. If you have questions, I see one of our two directors is here on the in the meeting, the directors of the pilot program. That's Wendy. So please, if you have questions about those, we encourage faculty as well as postdocs to consider applying. And I think I've already mentioned we've set the date for our symposium coming up next April, I believe. And you'll be getting more news about that. I don't see Rhoda on. So unless anyone has any other things about the ARC they want to point out, want to thank everybody. Have a great rest of your day.
Bye.
Thank you. Bye.
Thank you.
Bye-bye.
Bye.